Very good morning to everyone and those who are joined in a private chats and also in YouTube. It's our immense pleasure to welcome you all in this Altum platform. I am Divya here as a senior application scientist of Altum Technologies, joining along with Dr. Sonal Dubey ma'am, the reputed professor, and then Dr. Sonal from a College of Pharmaceutical Sciences from Dayanand Sagar University to given a very brief and then research interest of a QSAR. As soon as if you hear about a QSAR, lots of QSAR will comes in your mind. So ma'am had a very good and great experiences in developing the QSAR. Before starting the sessions, I would like to brief about a ma'am's profile. She has procured her PhD from a synthetic chemistry along for a medicinal interest and M.Pharm from an University of Pharmaceutical Sciences, Punjab University, and she is in a worked as in a very great experiences in various reputed colleges and pharmacy colleges. To her and accolades, she had more publications in international journals and national journals, and lots of achievements for in a drug discovery domain. Apart from that, she has designed some new kind of molecules for an optimization techniques, and compared to that, when you come to the academic, H-index journals are always been considered as a higher. So she had a 14 H-index and more than 562 citations to her accolades. And to addition to that, she is a principal investigator for an QSAR project sanctioned under a SIRS fast track by a DST Government of India. And she is also the co-principal investigator for a design and synthesis of QSAR. So when I say she is in, that shows how much she has been have an intense experience in developing the QSAR models. So I think this is more appropriate to have you in a platform, ma'am, to using your intense knowledge in the Altum platform. Let the participants get information from you using the QSAR application. By throwing your seeds, let many people also to gain this knowledge and come out with the QSAR applications. So thanks a lot and welcome you once again, ma'am.
Thank you so much, Dr. Devya, for such a nice introduction. I hope
So
I stand up to your talk about me.
definitely.
So,
So over to you, ma'am. Please go ahead and then we'll
thank you
connect you
so
in the Q&A
much.
session, ma'am. Thank you.
Sure. Thank you so much. So, first of all, I would like to thank Altum for inviting me for this talk, and in that particularly Dr. Divya and Dr. Sakshi. So thanks a lot for giving me this platform to share my knowledge and to let me present in front of you of one of the work which I have done on QSAR. So can I have the slides, please? Thank you. So, good morning to everyone. As Dr. Divya has told, I am Dr. Sonal Dubey. I am working as a professor, and today I am going to give you a brief talk regarding one of the work which we have done in our labs on HIV-protease inhibitors on amprenavir derivatives. So, we are going to look into this work, and along with that, we are going to first we are going to look at cheminformatics, that what is cheminformatics and how cheminformatics can be used in drug discovery. Okay, so drug design and discovery is the use of cheminformatics using QSAR for drug discovery. So the first part of my talk will be devoted to the cheminformatics, that what is cheminformatics, where it comes into picture in drug discovery and designing. And then the later part will be the case study of the amprenavir derivatives, which we have done for as a HIV protease inhibitors, which we have tried to work on them using QSAR approach. So let us begin. So let us begin from the beginning. As we all know that drug discovery is a very big process. takes around 12 to 15 years to come out with one drug in a market. So this drug discovery is starts from a disease. So when we want to do a drug discovery, what do? First thing we do is we try to find a target disease, the disease we want to target to make the drug discovery. So we will, this is a way which is rationally done, okay? So when we want to do a rational drug discovery, we always start with targeting the disease. Once we have targeted a disease, suppose we want to find an anti-cancer drug, so we will say, okay, we are targeting a disease, but that is very vague. Cancer has so many types, subtypes in it. So what do? We try to cut it down, short it down to a very basic level, like we want to do, say, for the breast cancer drug discovery or for lymphoma drug discovery, whatever the type of cancer we want to do. After we have selected that subtype of the category, then we have to find a target. What target we are going to uh, point out, we are going to address during our drug discovery. So our drug target is usually the place where our drug will go and bind and exerts its effect. So we start with the disease, we find a target. Once we have understood the biochemistry of the disease and we have found out what is the target for our disease, so we take that target into consideration and then we take the molecules which can go and bind to that target site. Okay, so we take the molecules and try to bind them to the target site. Then once we are able to get some results out of the candidates which we have, if some of them are showing positive effect, we go for preclinical studies. That is the studies on the animals for toxicity, for pharmacokinetics, as well as for activity. So we go for animal studies. After everything comes positive in the animal studies, the next part is you go for clinical trials. We all know there are 4 phases of clinical trial: phase 1, 2, 3, and 4. So after toxic preclinical studies, we go for clinical studies. And once everything comes out well, here as well, then the drug, your molecule becomes a drug molecule. Okay, till then it is your candidate only. Only when it passes the clinical studies, we make it a drug molecule. The chemical entity becomes a drug molecule. Okay, so this is the very basic which all of us as a medicinal chemist, biochemist, or the drug discovery development people, this is how we rationally go for drug design and discovery. So what are the targets we are looking for? The targets we see is, like, we have various targets. Targets can be receptors, they can be enzymes, hormones, the ion channels. It can be your nucleic acids, DNA, RNA. Those can be also your target. So if we see this pie chart, we can say that 45% of all the targets which we aim for are receptors. Okay, so receptors are a very big target in drug discovery. Then next comes the enzymes. So we try to make some enzyme inhibitors, or maybe something which activates the production of the enzyme or activates its activity. So next to the receptor, the next target is enzyme. Then comes the hormones, which we may want to target. Ion channels like potassium ion channels, calcium ion channels, sodium ion channels, channels like that. Then DNA and RNA, the nucleic acids, which makes a part of the gene therapy also. So we target them, and there are certain targets which are unknown, and there are around 7% of those targets for which we do not know what is the target exactly for that particular disease. So this is where the science still remains to be seen. So these can be the various targets which we want to aim for when we are going for drug discovery. If we see a drug discovery process, when you start screening around 1 million molecules, so you take a library, say you contain, in that library you have 1 million molecules, you do high throughput screening of them for a particular disease for which you have targeted. So you will find out that only half of them are coming out to be active. So only half will be there, which will be showing a little bit of activity, with which you can go further with. Okay, the rest of the half will be discarded there only. So out of the 1 million, half a million will say come active. Then what do you do with the active? You do some follow-up studies with it and you find out some hit molecules. So out of the 1,000 molecules, only 1 to 10 will come out as hit molecules. Okay, which are the ones which are going to hit the target, not the bullseye, but they are going to be somewhere around there. Okay, on the target board. These are your hit molecules. Now, out of these hit molecules, there is a very, you want to find a lead, which will be the leader in the series. Okay, lead is a molecule which will be like a, as the word suggests, leads the series. Okay, it's a leader in the series. So you can come up with only 0 to 3 leads when you start with 1 million compounds. So at the most, you will get 3 lead molecules. And when you further develop them, you may, if you are lucky, you will get 1 drug candidate. So this journey in itself, you can see, starting from 1 million and ending up with 1 molecule is a very tedious journey which your molecule has to travel. So out of this 1 million, you're looking for this one compound which is going to become a drug candidate, right? So we can see from here only how tedious the process is and how much of the energy, time, resources we will require to do this. Now, since the advent of the computational studies, like computers have come into the all fields of our life. So, drug discovery, we are taking the use of computers, we are making the use of computers to shorten this time to reach from here to here and to avoid failures. Okay, so how we are going to use computers? There are so many ways to use computers in drug discovery process. So what we are going to concentrate today is only on one aspect of it, that is the cheminformatics part of it, which is your QSAR. So there are so many ways which you can have in the other seminars, which will maybe follow it up here or elsewhere also. You can look up into that how we can shorten this process and reduce the failure rate and save the time and money so that we can come up with the one molecule which we are looking for. So taking help from the computers is what this generation has done very well. This and the previous generation has started with it, and now it is being carried out very nicely, and we are making full use of it so that we reduce our success rate. The success rate, we have to increase our success rate and reduce the failures. So why do so many compounds fail? Out of 1 million, only one compound is becoming a drug molecule. Why do we think that so many failures are there? Where are all these people, all these molecules going in between? What is happening to them? The failure is because of many different reasons. Around 40% of your molecule fail because of their pharmacokinetic problem. Pharmacokinetic problem is ADME problem, the problem related to their absorption, distribution, metabolism, excretion, or something. So this is your pharmacokinetic problem. 40% of them can be failing because of that. Then 30% of them, they fail because they lack efficacy. So at one point of the, like in preclinical trials or somewhere, they lack the efficacy which they had shown in your high throughput screening earlier. In HTS, they had shown that they were active, but when actually did in vivo on the animals, they lack the efficacy. So 30% are going to go there. Then 10% do not come ahead because they show adverse effects. Around the same percentage go out of the studies because of the toxicity problems. Okay, Then there can be some miscellaneous problem, which can be formulation related problem, that you are not able to formulate in a way you want to formulate it. Like you are not able to make a proper tablet of it, or you're not able to make a injectable form of it. So there can be some other miscellaneous reason. And finally, there can be some commercial reason. The interest of the company or the amount of the money which is being involved is not matching with the research inputs which are being carried out. So for commercial reasons also, many molecules get dropped down. So these are the main reasons of abandoning the development of any of a drug candidate. That these are the reasons a drug candidate is lost. Is there any chance, Divya ma'am or Sakshi, that I can get a pointer here, a cursor or something? Okay, let me continue it like this only. So we are going to see where the cheminformatics is coming into picture. When we have found a disease and we have found a target, the cheminformatics is the portion which is covering the region of finding the drug target and finding the drug molecule. So this is the square in which we can use cheminformatics. This is the place where cheminformatics will come into picture. Okay, so how we can use cheminformatics here? For that, first thing we should have to do is we have to understand what is cheminformatics. This term cheminformatics, it was introduced in 1990s by F.K. Brown, and it first appeared in the scientific article in 1998. So it was in the late 90s when this term was officially being used in the scientific article, cheminformatics. So by definition, cheminformatics is a field of information technology that uses computer and computer programs to facilitate the collection, storage, analysis, and manipulation of large quantities of chemical data. So that is what is the cheminformatics about. The use of computers to facilitate collection, storage, analysis, and manipulation of large quantities of chemical data. So cheminformatics is not one thing. It is a combination of different categories of science. So you cannot say that only one sciences will deal with cheminformatics. There are so many scientific disciplines which get together to make cheminformatics, like library science, chemistry, computer science, toxicology, biology, drug discovery, medicines, text mining, and informatics. So all of these together will make cheminformatics. So all of you must have heard of the word bioinformatics. There is a little bit of difference between bioinformatics and cheminformatics. What is that difference? Bioinformatics, it deals with the biopolymers, a sequence of repeating units. So biopolymers are usually a sequence of repeating units. Cheminformatics it deals with the chemicals. So we do not have any sequence and there are no repeating units in it. So this is a very small amount of difference between bioinformatics and cheminformatics, that this is a sequence, repeating sequences there is no repeating sequence in the cheminformatics because we are dealing with the individual chemical structures. So when we are talking about cheminformatics, we will be dealing with the molecular descriptors and the method to compare these descriptors. There are various public libraries which are available, which will have your chemical databases. So many of them are there. I have listed a few. There are so many more here also, which will be used in the, which contains the chemical databases. So if you want chemical databases, you go to any of these, or 3-4 of them you explore, you will come to understand that how you have to get your database, because you can search depending on the nucleus you are looking for, or if you're looking for the analogs of a particular structure, you can find those analogs as well. Or if you want with the activity that the compounds which are active for one particular protein, so you can get the database search from there also, and the chemicals will be shortlisted for you. Okay, so you have you have large number of these kind of chemical databases. So these molecular modeling or the cheminformatic methods, they strongly depend on one thing, which is quantitative structure activity relationship, QSAR analysis. Okay, the basic principle of computational method relies on QSAR model, their descriptors, statistical phenomena towards the molecular structure. So this is what the basic principle of computational methods using cheminformatics depends on. What kind of QSAR model is obtained, what are the descriptors which are present in it, and what are the statistical parameters which have come out of this study. So this was the very basics about the cheminformatics and QSAR. So let us take up one case study which we had done in our lab previously. So we have been working for a long time on HIV inhibitors. So this particular case study is on HIV-protease inhibitors. So one very useful drug as HIV-protease inhibitor is amprenavir. So we have done some work, QSAR related work on amprenavir derivatives using different softwares. So that is what I'm going to present now in this remaining part of the session, that what was the work which was carried out and what is the result which we have got using QSAR. So before we begin with the QSAR, I would just like to tell you in brief that what is protease market looks, what is protease inhibitor and how it works and how the inhibitors have come into picture. So HIV, as we all know, it causes AIDS. So HIV is a lentivirus which belongs to retrovirus category. This is the retrovirus. This is the structure of the HIV-1 virus. This virus goes inside the cell, okay, through the cell membrane. Inside the cell it goes. Now this virus is transmitted as a single-stranded enveloped RNA virus. This is a RNA virus. So you do not find DNA in it. Since it's a retrovirus, it is an RNA virus. Upon entry, the RNA genome is converted to double-stranded DNA. Okay, so the conversion of RNA to a double-stranded viral DNA is done with the help of an enzyme known as reverse transcriptase. All right, so there is an enzyme which is called reverse transcriptase, which helps in the conversion of viral RNA into viral DNA. Next thing, this viral DNA enters the nucleus and then it gets itself integrated with the DNA of the humans. So when it gets integrated, I hope you can see the color difference between these two. This is the gray color, which is the human genome, and this is the blue color, which is the viral genome. So this is the DNA part of the human, the gray and the blue is the viral part. So this virus DNA then integrates itself with the human DNA. So the enzyme which works here for the virus is integrase. As the name suggests, it is integrating. It is integrating the DNA of the virus into the human genome. So that when the human genome replication is taking place, this DNA replication is taking place, along with that viral DNA also gets replicated. And the virus increases in number. Okay, so we see that there are two enzymes so far which have come into picture, reverse transcriptase and integrase. Now, after integrase has integrated and the mRNA has been formed, the viral mRNA has been formed, there is another enzyme which is protease, which is required for the maturation of this into a full-fledged virus, which can be sent out and infect the other cells. That's how the infection propagates. Okay, so the RNA is being multiplied here, replicated here, and this replicated RNA is giving rise to multiple viruses, which will then move out of the cell and infect the other cell, and the infection propagates, the disease propagates itself. So as of now, inside the nucleus, inside the cell, we have three enzymes which can be targeted for HIV: reverse transcriptase, integrase, and protease. All of them are crucial for this virus to multiply. Another one is the entry inhibitor, which is CCR5 and CXCR4 receptor inhibitor. So if they do not let the virus come and bind to the envelope, it will not enter. Okay, so these are the 4 possible places where you can stop the HIV infection to propagate itself. So our interest today is of the HIV protease inhibitors. So we are trying to find some molecules which will go and bind to this enzyme and inhibits the activity. If they inhibit the activity of this protease, what will happen is it will not let this mRNA to mature into a full-fledged virus. And if that is not happening, you're stopping the infection there. Okay, you're stopping the disease there. So amprenavir is one such molecule which acts as a protease inhibitor. Now this is the structure of the HIV-1 protease. This is the enzyme. It is having 2 flaps. So this side of the right side of the molecule and the left side of the molecule. So it's a similar kind of a structure having 2 flaps. Now this dotted side in the center is your active site. Okay, so this 25 to 29 amino acid sequence is your active site for HIV protease. So whenever you want to target a disease, target a molecule which is a protease inhibitor, you have to target something which will go and bind here in the active site. So what work we had undertaken? Sorry. This structure is not coming here. We have taken a series of cyclopentyl methyl substituted phenyl sulfonamide 5-hydroxypropyl acetamide derivatives. So this is the structure of the amprenavir. I don't know, this structure is not coming here right now. This is the structure of this molecule which I have written here. Is supposed to be here. So anyway, so we have taken this series of the molecule and they have related structure to the amprenavir, and their activity, these molecules were taken from literature and their protease inhibitory activity was also reported. So we have selected same series of molecule and from different studies on which the protease inhibitory activity was already done. So we have taken 96 compounds for the study and divided into the training set and test set molecule. So we have taken 80 to 20 training to test ratio. You can also take 75 to 25. So 80 to 20 is the minimum which at least which you should take. You can take more of the test set also. 75-25 is also fine if you can, if you want to take that way. So we divided our compounds into training set and test set, and the values which we have taken is the log inhibitory values for the protease inhibitory activity for the study. So we have basically done QSAR using multiple techniques. We have used 3 different softwares and we have tried to compare the result and try to come out with certain molecule which has the probability, certain substitution in a molecule which will have a probability of being a better protease inhibitor. Okay, so that's the work which we have done. So to do QSAR, we need to have certain molecular descriptors. Molecular descriptors are basically the physicochemical parameters of the molecules, which either some of them you can also calculate actually for the molecule, or if you give the structure of the molecule to the software. There are many softwares available in market which are free as well, which can give you the different descriptors for the molecule. So there can be a number of different descriptors. Under each category, you can have 50 to 100 sub-descriptors. So molecular walk count, constitutional descriptor, charge descriptor, functional group, gateway descriptor, 3D most descriptors. So all these descriptors will have further subcategories. And when you give one molecule to the compound, depending on the software which you're using or the facilities which you have, you can calculate up to 1800 to 2000 different descriptor for one single structure. Okay, so you draw your molecules and give it to the system, to the software, and they calculate the descriptors for you. So I don't know why. Sakshi,
Yes, ma'am.
Sakshi, actually these slides were supposed to come like one after the other on the press of the key. Is this facility available, or should I just go ahead like that?
You can only do, ma'am, actually.
No, because it's coming all together. I have designed them that one at a time, one tab will come. I think I'll explain it like this right now.
Yes, ma'am.
Because when I made it in a PowerPoint, it was coming one after the other on the press of the key, but now it is not coming like that. Okay, let me try to explain it this way only. Okay, so what we have done, the first software which we have taken is called CODESA. So it's a CODESA software. We have drawn the structures and using ChemBio Ultra. Then we have given those structure to a descriptor calculating software, which is Dragon. So Dragon calculated around 1664 molecular descriptors per structure. For each structure, around 1664 molecular descriptors were calculated. So we have taken all those descriptors for all the compounds which we have taken for the studies, and then we had given this input of the descriptors to another software, which is CODESA, which is your QSAR software. So QSAR is a software which does QSAR based on principal component analysis and heuristics. So principal component analysis is you divide your compounds based on the different principal components which are present in the compound, the different descriptors which you have given. And this green dot, if all of you can see, this is of the active compound, amprenavir molecule itself. So these are the molecules, the other red dots in this quadrant of the principal component analysis are the ones which have the similar characteristic like your drug molecule. So all these compounds which are having the red dots are in this quadrant are the ones which have the probability of showing activity, good activity. These are the ones having good activity. So these are the ones which are having, these are the descriptors which will show you which are the ones which are responsible for the activity. So these names of the descriptors are there if you can read out. So these are the descriptors which are going to have a positive impact on the activity of the molecule. So principal component analysis will give you the bunch of those descriptors which are good for activity. So once after, and the other one is the heuristics, which is your MLRA, multiple linear regression analysis. So using all the descriptors, multiple linear regression analysis was performed by the software, and then you got various QSAR models. So these equations are your QSAR models. So activity is given in terms of some constant value multiplied by the descriptor. Then if the descriptor is positively contributing, you will have plus sign in front of that. If the descriptor is negatively contributing to the activity, you will have minus sign in front of it. So activity will be, it's a linear relationship. So the equation which has come out is y is equal to mx plus c. So which is a straight line equation. So c is your constant, which is the first number in this equation. In all the equations, you will have one constant, which is the first number, and then mx are the rest of it. Okay, so from this activity equations, which is your QSAR equations, you have to find out the one which is the best one. So how do we choose which is the best equation which has come to us? we have to see the R square and Q square values. Okay, that is the utmost importance, R square and Q square. So what is your regression coefficient and what is your cross-validated R square, Q square value? So R square, anything above 0.8 or 0.85 is good. So as you can see, all of the equations have 0.9 and above. So R square is good. So now let us see about with the Q square values. So 0.6 seems to be a good Q square value to have. So all of them are falling in that category. We have chosen this because of the statistical parameters F value, S square and P value, which is optimum for this equation. So this equation, which is in the red color, we have chosen this to be the optimum one. And because we do not want anything to be out and out to, like, only we cannot look at R square values, we cannot only look at the Q square values, we have to look for all the other parameters and come out with the optimum equation, which will have the probability of giving you a better result. So with this QSAR model, we came up, we have chosen the one which is there in the red one, and we saw the plot of it. So calculated versus the experimental value. So we have your equation which you have selected, you ask that equation to calculate the activity for the compounds which I've given. You actually have the experimental value and you plot the graph. So you can see that most of the points are lying around the central line. So this seems to be a good R square and good equation. This is in the box is the residual graph, the graph of the residuals. So we use this equation to see the test compounds, how well they were predicting the activity of the test compounds. So these are the few of the best compounds whose values were predicted using this equation. So you can see the results are quite comparable. So experimental value and the calculated value are quite comparable to each other. So that means we have chosen the right kind of equation for our QSAR using CODESA. So this prediction will tell you whether you're choosing the right kind of equation or not. So you have to first go with the statistical parameters, and then you put your calculated value of the test series. You ask the equation to calculate it for you, and you compare it with the experimental value. You will come to know whether the equation is right for you or not. If it's not giving good results, you can try the other equation as well, which is nearby to the first one. So this was the first software which we have used. The second one we have used is Topomer Compa. It is a Tripos USA's software. So we just wanted to compare the activity of different softwares, QSAR potential, QSAR result potential of the different software which are available. So we used Tripos also. We did the model making using the Siebel base, and then the usual procedure to do the QSAR. So new Topomer Compa was made, and we got a 3D QSAR model. Now, when you do Topomer Compa, you usually get models in this kind of a result. Results are usually this kind, which is you get different fragments. Our molecule had a basic central structure, and we had two R1 and R2 fragments in it. So the R1 fragments contour plots were made, and R2 fragment contour plots were made. So this part of the result is showing the green and yellow shields are there. They are showing the steric vectors. So green means this if you have a steric group over here, which is favorable. Yellow means if you have a steric group over here, it is not good for the activity. So these are the places where you have to see in the molecules where you need to put the steric group and where you should not put a steric group or a bulkier group. Similarly, electropositivity and electronegativity, if you see, the blue color shows that electropositive is good for the activity, and red shows that electropositivity is bad for the activity. So what kind of groups you're going to put in this substitution, you can come to know from these kind of Topomer Compa plots. So the first one, this one was for the R1 fragment, and this one is for the R2 fragment of the molecule. So these are basically the two end substitution in the molecule, which we have studied. So again, the steric parameter and the electrostatic parameters, where we should go for it and where we should not go for it. So these topomer-compa models can also be used to calculate the activity of the test compounds. Again, as you can see, the results are quite comparable. Okay, barring a few, the results are quite comparable. And this is the predicted versus calculated activity graph which has come from compa. So if it is coming in a straight line and more compact this line is, the better your model is. Okay, so how do you see whether a model is good or not? The thinner this spread is, the better this model is. So if all of them are falling on the same line in the same group together in a straight line, then the better the model is. So using topomer compa, we got the R square value of 0.81 and Q square value of 0.608, which is a good model. Okay, R square 0.8 and above, I told you, is okay, but this is the best we got from topomer compa. Then we have used another software, which is Biograph Suite. This Biograph Suite is software which is developed by University of Basel, Switzerland. So we had worked with them and we got the software from them. We have purchased the software. This software has the ability to do 3D, 4D, 5D, and even 6D QSAR modeling. Okay, so this was the specialty of the software, that we were not limited to 3-dimension. We did 4-dimension, 5-dimension, and it takes care of 6-dimension as well. And the final results which you get is your QSAR model, which is your 6D model building. So what are these different dimensions other than the 3 dimensions, which 3 dimension we all understand, X, Y, Z axis of a molecule, which we consider for the study are the 3 dimensions. Now, what is the 4th dimension, 5th, and the 6th dimension? The 4th dimension which this software uses are the different conformation, their orientation, and protonations. So this is the 4th dimension which the software is taking into account. Each molecule which you have can exist in various conformations. So all those conformations are accounted for. Then the orientation, the way they are oriented. In one orientation, there can be different conformations, right? So similarly, protonation state can also vary. It depends on the solubility, it depends on the pH. So what is the protonation state, whether it is having a hydrogen bond acceptor, donor, or hydrogen flip-flop is there, what kind of protonation state it is in. So that becomes the 4th dimension of the QSAR. The 5th dimension, it allows the simultaneous evaluation of 6 different induced fit protocols. So the 5th dimension is the different induced fit models which it can generate to fit into that space. And the 6th dimension which QSAR allows us is the consideration of solvation model, the various solvation models which occurs because of the presence of water in the body. When the molecule is actually going in your biological system, it is surrounded by the aqueous environment. Okay, so that solvation state is taken care by the 6th dimension. So all in all, this QSAR looks very good. It's taking into account all the different possibilities which are there. So we try to use it. So what we have done first is we have done a 4D alignment. 4D alignment means not only the alignment of the molecules, but even of the conformations which were obtained, various conformations and the orientations which were obtained of each single molecule. So we got this kind of output in that. So they call it as QSAR. Then we have done their QSAR, which consisted of 2D, 5D, and finally the 6D QSAR. So you get a dual shell representation. The output comes in this kind of a representation. So you can see this is a mesh kind of a representation, and there is this other kind of a representation. So this is a wireframe, which is the outer shell. The inner shell is depicted as the water wireframe, and the outer shell is depicted as a surface fill model. This is the surface fill model. So two shells. The Raptor model, it is showing the dual shell model. And when you compare the activity of the predicted versus calculated, you can see that this is very much in the line and very much within the limits of the regression. So the line is straight and the points are falling within the same region in that line. Okay, so this is your predicted versus experimental activity of the inhibitory values. So when you do the 60 QSAR, you see this is the structure of the receptor which you have taken, the protease receptor which we took from PDB, along with the this is the amprenavir derivative inside. These dotted things you can see, this is the amprenavir itself inside. And this is your monorepresentation of the surrogate for the series of protease inhibitors which we have taken for the studies. So this protein is acting as a surrogate, and inside the surrogate your molecules have been aligned together. This is a visual representation of the thing. Okay, but this is not docking. This is, in the end, it is a QSAR only. So what this thing is depicting, when you have your protein and you have tried to fit your structure inside the protein, it is not showing you the normal molecular docking bindings, that this is the binding energy, or these are the hydrogen bonds, or these are the amino acids involved. The results is not coming in terms of that. The result is coming in the terms of these dots, which you can see on this wire mesh mapped. Okay, these dots represent, the different colors of the dots represent different things about the molecule. So like the red one, they tell you that where should be the positive charge of the salt bridge. The blue one will tell you the negative charge. The green one will tell you that it is a hydrogen bond donor. The yellow ones are the hydrogen bond acceptors. Okay, light browns are positively charged hydrophobic molecules. So this is telling you about what is required out of all the molecules which are present, the ones which are showing the binding inside. What is the property of those things which are binding? So which are the ones which are required for it? Okay, so this map is going to give you a very good data to understand about your study, that what you have understood from the test set and training set, and what is the how you can imply these things into your further studies, how you can work on this for the further development of a better compound. The aim of all this QSAR or cheminformatics or bioinformatics or whatever we do is to design a better drug molecule, to design a better structure which will have a probability of becoming a drug molecule, right? So this kind of representation is very nice when it is going to tell you that which are the points which you should take for the further development of your compound. So these are the statistical parameters which will tell us that the model which is developed, the 60 model which was developed, is good or not. So again, the comparison between the experimental and the predicted binding affinities, and you have to do scramble tests to see whether the model is good or not. So the scramble test showed good results. This is the scattered plot. It is showing you this cross-validated R square versus the predictive R square. So they are all in the one region, which is again a good sign. So these are all statistical parameters with which you decide. So if we compare the biograph results with the topomer compa results, we can see that the results are comparable. Q square values, R square values are comparable. So both of them are giving comparable results, and based on these results, what we have done is, in our study, we have designed the different substitutions which can be made in the amprenavir molecule so that the probability of getting an active compound is more, so that we can get a better derivative if we go ahead with these kind of substitutions. These are not the substitutions which are initially present in amprenavir. Okay, these are the ones based on the study which we have suggested that if you go with these kind of substitutions on the amprenavir derivatives, we can there is a higher amount of possibility that we end up with an active compound, because there is no 100% guarantee when you do computer aided drug design. There is no 100% guarantee that if you're going to make this, it will become a drug molecule. But the probability of getting a positive result or going to the higher level in the drug discovery line becomes better, and it is a more rational approach to do the work. So it is not the only, but it has, your work has to go via computer aided drug design to the drug discovery. So it is now this computer aided drug discovery is an essential part of the drug discovery. We cannot leave it on side and go with our hunch and start making compounds. We have to use computer aided drug design and then go for the chemical modification and then see the activity and then proceed with the preclinical and clinical trials. So the probability of you getting a success becomes higher, and it's a more rational approach, and you will save, in the process you're going to save a lot of time and money which otherwise would have been wasted for no good reason, because you do not know why you're synthesizing what you're synthesizing. So if you do some kind of a computer aided work, you can be more rational for your work. You can have a better understanding of what you are doing and why you are doing. So that is the whole idea behind the computer aided drug design. So these are some of the references which we have taken for this work. And thank you very much. With this, I end my talk.
Thanks a lot, ma'am. You have given a very good insight about this information of QSAR. And before that, there are few questions are there.
Yes, ma'am.
I would like to ask. Ma'am, can you please let us know where to get a data for performing the QSAR?
Okay. Okay, see, the data from QSAR, there can be two ways to get it. One is you have your own lab which does the work, and you have done the synthesis and you have done the activity on particular series, and you have synthesized say 100 to 200 compounds in that series, and activity has been done for those compounds. Then you can use your own data for the QSAR studies. If not, if you have not done that, you can take it from the literature. Okay, go to the literature, find the literature which is relevant to your work. And people have done synthesis. There are so many journals out there in which people will do the synthesis and do the activity, and it will be published. The only thing which you have to remember while you're collecting the data is your basic nucleus should be same and the activity testing pattern should be same. So whichever method has been adopted for the activity testing, it should remain same throughout the articles. If you're combining 2-3 articles, suppose you're getting 20-25 compounds only in one article, another article is also giving you 20-25 compounds. So you can merge them only if the way they have been tested is same, and the basic nucleus should also be the same. So that is the way you can go ahead.
Yeah, thanks a lot, ma'am. I think this is very important where the people will make a mistakes actually. They will try to find out, if I say HIV, they will try to find out for the different HIV molecules, but not pertaining towards a single drug target. I think this is very important. Thanks a lot, ma'am. And we have few more questions also.
Yeah.
I think descriptors are the task in QSR. And there is a question, is there is any way to say we should select a specific descriptor for a study?
Yeah. So when you first you have to give your molecule to the software to calculate the various descriptors, and then the software will only tell you which are the important descriptors which are contributing to the activity. Okay, so when your results will come, the QSAR equation will come. It will, as I have shown you in my slides also, there are certain descriptors which will contribute positively towards activity, and there are certain descriptors which will contribute negatively towards activity. So looking at them, you can come to know that which ones are good for activity and which one should be taken and which one should not be taken. So look at the QSAR equation. The positive ones are the ones which are contributing towards the activity. So look for those ones.
Yeah, that sounds good, ma'am. I think this is the one people will make a mistake. And there is also again questions. What are the applications of QSAR?
See, as I've told you, like we have, after doing this QSAR, we have come up with certain substitutions which you should make in the basic molecule. Okay, so you can suggest based on that, that what all different substitution you can propose for the future synthesis in your lab. Okay, so that is what is the application of QSAR. You have to propose that these modifications can give you better results. Or what you can do if you have a QSAR equation, not in a model form, if you have in an equation form, you can predict without synthesizing only. You can calculate the descriptors for your compound and put those in the equation and see whether if you synthesize this particular compound, will the compound be active or it will not be active. Because activity will be, as I've told you, it is a straight line equation. So y is equal to mx plus c. So you have a constant and then you have your descriptors multiplied by some number, which will be contributing towards the activity. So you have to, without synthesizing only, you can predict whether your compound will be active or will not be active. If it is giving you a positive result, you should be more enthusiastic to go for the lab work and do the lab work.
Yeah, that's great, ma'am. I think this is the main application of the QSAR. And if there is any more questions? Fine, ma'am. I think that sounds very good, and we are really great to have you here. And
thank you so much,
so
Dr.
much of
Divya.
yeah, because so much of information about QSAR, because it's a very hard topic to understand in cheminformatics. Usually, people will try to find out where the problem and what method have to select, and coming to the descriptor problem. So all these things you have beautifully explained, and that is from the right of drug discovery process and going ahead with what are the steps in the drug discovery process and importance of the drug discovery and why QSAR and also what QSAR. To an extent, you are briefed in detail about the positive and the negative contribution of this descriptors, and using that how we can predict the activity and value of the unknown compounds also. So put together, it's in a whole essence of QSAR is given by you. And similarly, you have given brief about 2D, 3D, and 4D, 5D descriptors. And I hope this will be you helpful for all the peoples and using different softwares. And even in the BioVR Discovery Studio also, we have a 2D, 3D, and more than 1,000 descriptors you can able to predict and to enhance your results. And I think this is on a full-fledged of QSAR. Using this, one can do any kind of QSAR, I believe so. So again, thanks a lot, ma'am, for joining us. It's a pleasure to have you on a platform to enrich your knowledge about QSAR. So as
Thank
from
you so
Altum
much.
yeah, ma'am, from the Altum Technologies, I thank you once again, ma'am. Thanks a lot, ma'am.
Thank you. Thank you. And I thank Altum Technology to give me the opportunity to share my things. Thank you so much.
Thank you, ma'am. Thank you.
Thank you.